Hello and welcome dear students. Today we are going to discuss the topic genetic code. The main objectives of today's lecture are to know about the basics of genetic code, to study the purpose and principles of genetic code, to know about the features or characteristics of genetic code, to know the exceptions to the genetic code and mutations in the genetic code, and to study the benefits of genetic code. Dear students, let's first start with the introduction to genetic code. Our genetic information is stored in our chromosomes on sections called genes, and each gene code uses the 4 nucleotide bases of the DNA, that's adenine, cytosine, guanine and thymine, in various ways to spell out the 3-letter codons that specify which amino acid is needed at each position within a protein. So, decoding messages contained in DNA is a key step in gene expression, in which information from a gene is read out to build a protein. A 4-letter language is translated to 20-letter language during protein synthesis. There should be a specific relationship among the 4 bases of DNA and sequence of 20 amino acids in the protein. Codons are the units of information in mRNA. They are made up of 3 consecutive nucleotides, each of which can be one of the 4 bases, that's uracil, cytosine, guanine or adenine. 64 different codons result from all the combinations. So, 20 amino acids are coded through the phenomenon of translation, that is the protein synthesis. A 21st selenocysteine is coded in some organisms in particular situations, and a still very rare 22nd amino acid, that is pyrrolysine, has recently been discovered. Dear students, let's now discuss the purpose of the genetic code. Genetic code can be defined as the set of certain rules using which the living cells translate the information encoded within the genetic material, that is the DNA or mRNA sequences. Genetic code refers to the instructions contained in a gene that tell a cell how to make a specific protein. A gene is a portion of the DNA that codes for a trait or a character. Genetic code is responsible for our traits or characteristics. Our genotype is our genetic code of our cells. The source of different characteristics or characters of a living being is coded in the genes. So, genetic information is thus passed from one generation to the other in the form of code. It decides the traits of an organism, whether it is a bacterium or a human being. The entire life process is controlled by the genetic code one possesses. Genes that provide instructions for proteins are expressed in a 2-step process. In transcription, the DNA sequence of a gene is rewritten in RNA. In eukaryotes, the RNA must go through additional processing steps to become a messenger RNA or mRNA. In translation, the sequence of nucleotides in that mRNA is translated into a sequence of amino acids in a polypeptide, that is the protein chain. Genetic code is the mechanism by which genetic information is stored in living organisms. So, the code uses sets of 3 nucleotide bases, that is codons, to make the amino acid that in turn constitute proteins. So, we can also define the genetic code as the set of correspondences between the codons of mRNA and the amino acids of the proteins produced on the ribosomes through the translation of these mRNAs. The genetic code is of 2 types. The genetic code can be expressed as either RNA codons or DNA codons. So, RNA codons occur in messenger RNA or mRNA and are the codons that are actually read during the synthesis of polypeptides, that is the process called translation. Dear students, Let's now understand what are codons. Cells decode mRNA by reading their nucleotides in groups of 3 called the codons. So a codon is a sequence of 3 nucleotides which together form a unit of genetic code in a DNA or RNA molecule. The concept of codon was first described by Francis Crick and his colleagues in 1961. During the same year, Marshall Nirenberg and Heinrich Matthey performed experiments that began deciphering the genetic code. They showed the RNA sequence UUU specifically coded for the amino acid phenylalanine. Following this discovery, Nirenberg, Philip Leder, and Hargobind Khorana identified the rest of the genetic code and fully described each 3-letter codon and its corresponding amino acid. So codons are the units of information in mRNA and are made up of 3 consecutive nucleotides, each of which can be one of the 4 bases: uracil, cytosine, guanine, or adenine. 64 different codons result from all the combinations. So genetic code actually allows DNA and RNA sequences to be decoded into the amino acids of a protein. The genetic code is the set of rules used by living cells to translate information encoded within genetic material, that is DNA or RNA sequences of nucleotide triplets or codons, into the proteins. Here are some features of the codons. Most codons specify an amino acid. 3 stop codons mark the end of a protein, that is the termination. 1 start codon, that is AUG, marks the beginning of a protein and also encodes the amino acid methionine. Codons in an mRNA are read during translation, beginning with a start codon and continuing until a stop codon is reached. mRNA codons are read from 5' to 3' and they specify the order of amino acids in a protein from N terminus, that is methionine, to C terminus. An important point about the genetic code is that it's universal, meaning that the set of correspondences between the codons and the amino acids is highly conserved in all the organisms. That's with minor exceptions, virtually all species from bacteria to humans use the same genetic code. The information transferred or passed from the parent generation to the offspring is called the genetic code. The process begins at the cellular level when the genes get split into the parental reproductive cells and then get united to form a hybrid set of genes during fertilization or reunion of gametes. The complete set of relationship among amino acids and codons is said to be genetic code, which is often summarized in a tabulated form. Dear students, let's discuss the genotype. The genotype is a set of genes in the DNA which are responsible for the unique trait or characteristics to make your phenotype, whereas the phenotype is the physical appearance or characteristic of the organism. Thus, we can find the human genetic code with the help of their genotype. Organisms may look the same but still may not have the same genotype. One can determine the genotype by biological tests. On the other hand, the phenotype is determined by an individual's genotype and can be expressed as genes or by the visible traits such as hair color, eye color, body shape, height, etc. In biology, a gene is a part of the DNA that encodes some traits or characters. The precise arrangement of the nucleotides, each composed of a phosphate group such as sugar and a base in a gene, can differ between copies of the same gene. Thus, gene can exist in various forms across the organisms. These different forms are popularly known as alleles. Also, the exact as well as a fixed position on the chromosome containing a peculiar gene is termed as locus. A diploid organism may inherit two copies of the same allele or may inherit one copy of two different alleles from their parents. So, the individual inheriting two identical alleles is termed as homozygous at that locus, whereas if they possess two different alleles, then their genotype will be classed as heterozygous for that locus. Alleles having the same genes are either autosomal dominant or recessive. The subsequent combination of alleles that are possessed by the individual for a specific gene is their genotype. Dear students, let's now discuss the properties or characteristics of the genetic code. The genetic code has certain properties like it is triplet code, it is comma-less code, it is non-overlapping code, it has polarity as well, it is degenerate code, it has a start and stop codon, and it is non-ambiguous and universal. So, dear students, let's discuss them one by one in detail. Genetic code is triplet code. A codon or a code word is defined as a group of bases that specify an amino acid. There is a strong evidence which proves that a sequence of 3 nucleotides codes for an amino acid in the protein. That is, the code is triplet. The 4 bases of nucleotide, that is adenine, guanine, cytosine and uracil, are used to produce 3 base codons. The 64 codons involve sense codons that specify amino acids. Hence, there are 64 codons for 20 amino acids, since every codon for one amino acid means that there exists more than one code for the same amino acid. A 21st selenocysteine is coded in some organisms in particular situations, and a still very rare 22nd amino acid, that is pyrrolysine, has recently been discovered. So, each codon thus has 3 bases producing 64 codons. Out of these 64 codons, only 61 are capable of producing amino acids. The rest 3 codons are used as stop codons during the process of translation. 64 different codons result from all the combinations. 20 amino acids are coded through the phenomenon of translation or protein synthesis. Dear students, let's now discuss the genetic code is comma-less. The triplet codes are arranged one after the other on mRNA molecule without any gap or space, and therefore genetic code is considered as comma-less. It means that there is no room for punctuation in between, which indicates that every codon is adjacent to the previous one without any nucleotide between them. Dear students, let's now discuss that genetic code is non-overlapping code. The genetic code is non-overlapping. For example, a sequence UGGA UCGAU is read as UGG AUC GAU rather than UGG GGA and GAU, etc. The code has no punctuation, so no base serves as a comma between groups of bases. Therefore, the code is read sequentially 3 bases at a time. The code is read sequentially in a group of 3, and a nucleotide which becomes a part of the triplet never becomes part of the next triplet codon. For example, 5-UCU 3- codes for serine and 5-AUG 3- codes for methionine. Dear students, let's now discuss the genetic code is having polarity. The genetic code has polarity, that is, the code is always read in a fixed direction, that is in 5-3- direction. Therefore, genetic code has a particular direction of reading that message. Reading from the opposite direction will code for some other amino acid. Each triplet is read from 5- to 3- direction, and the beginning base is 5- followed by the base in the middle, then the last base, which is 3-. This implies that the codons have a fixed polarity, and if the codon is read in the reverse direction, the base sequence of the codon would reverse and would specify two different proteins. Dear students, let's now discuss that genetic code is degenerate. Although each codon is specific for only one amino acid or one stop signal, the genetic code is described as degenerate or redundant because a single amino acid may be coded for by more than one codon. So every amino acid except tryptophan, that is UGG, and methionine coded by AUG is coded by various codons. That's a few codons are synonymous, and this aspect is known as the redundancy or degeneracy. The degeneracy of amino acid coding is one of the most crucial and enigmatic aspects of the genetic code. As we know, there are 64 triplet codons and only 20 amino acids. Incorporation of some amino acids must be influenced by more than one codon. Only tryptophan and methionine are specified by single codons. All other amino acids are specified by two. For example, phenylalanine is specified by UUU or UUC, to 6 like arginine, which is coded by CGU, CGC, CGA, CGG, AGA or AGG codons. These are called degenerate or redundant codons. In degenerate codons, generally the first two nitrogenous bases are similar while the third one is different. As the third nitrogenous base has no effect on coding, the same is called Wobble position, as Wobble hypothesis discovered by Francis Crick in 1966. Dear students, let's now discuss that genetic code has a start and stop codon. As we know, the genetic code is degenerate, that's more than one codon can code for a single amino acid. Due to this, of the 64 codons, 61 codons code for the 20 amino acids. There are two punctuation marks in the genetic code called the start and stop codons, which signal the end of protein synthesis or translation in all organisms. Generally, AUG, which codes for methionine, is the initiating or start codon. The polypeptide chain starts either with eukaryotes, that is methionine, or prokaryotes, N-formyl methionine. On the other hand, UAG, UAA, and UGA are called termination codons or stop codons because they are not read by any tRNA molecule and they never code for any amino acid. These are also known as Ocher, Ember, and Opal. Dear students, let's discuss now the genetic code is non-ambiguous and universal. Genetic code is interrelationship between nucleotide sequences of DNA or mRNA and amino acid sequences in a polypeptide or protein. It is mRNA sequence containing coded information for one amino acid and consists of 3 nucleotides. One codon codes for only one amino acid, hence a genetic code is unambiguous and specific. Only GUG is the ambiguous codon. It normally codes for valine but at initiating position as well as codes for methionine. This is an exception to the genetic code. Otherwise, genetic code is non-ambiguous, which means a specific codon will only code for a particular amino acid. Also, the same genetic code is seen valid for all the organisms, that is, they are universal. Therefore, unambiguous code means that one codon codes for only one amino acid. For example, AUG codes for only methionine. Universal code means that codon and its corresponding amino acid in the protein are the same in all organisms. For example, bacteria to humans like UUU codes for phenylamine. Dear students, let's now discuss the collinearity of the genetic code. Both polypeptide and DNA or mRNA have a linear arrangement of their components. Further, the sequence of triplet nucleotide bases in DNA or mRNA corresponding to the sequence of amino acids in the polypeptide manufactured under the guidance of the former. Change in codon sequence also produces a similar change in amino acid sequence of the polypeptide or the protein. Dear students, let's discuss the cistron polypeptide parity. Portion of DNA called cistron, that is gene, specifies the formation of a particular polypeptide. It means that the genetic system should have as many cistrons genes as the type of polypeptides found in the organism. Dear students, let's discuss now the exceptions to the genetic code. The genetic code is universal since similar codons are assigned to identical amino acid along with similar start and stop signal in the majority of genes in microorganisms, plants, and animals as well. However, a few exceptions have been discovered, and most of these include assigning one or two of the stop codons to an amino acid. Apart from this, both the codons GUG and AUG may code for methionine as a starting codon, although GUG is meant for valine. This breaks the property of non-ambiguousness. Thus, it can be said that few codes often differ from the universal code or non-ambiguous code. Dear students, let's now discuss the different codons. In paramecium and some other ciliates, termination codons UAA and UGA code for glutamine. Dear students, let's now discuss the overlapping genes. In bacteriophage, 5*174 has 5375 nucleotides that code for 10 proteins which require more than 6,000 bases. 3 of its genes, E, B, and K, overlap other genes. So nucleotide sequence at the beginning of each gene is contained within gene D. Likewise, gene K overlaps with genes A and C. A similar condition is found in SV40. Now let us discuss the mitochondrial genes. AGG and AGA code for arginine but function as stop codon in human mitochondrion. UGA, a termination codon, corresponds to tryptophan, while AUA, codon for isoleucine, denotes methionine in human mitochondria. Further, the exceptions to genetic code can be summarized in the form of a table given here. Even these exceptions prove the rule of common descent, as major groups of organisms from mammals and plants have the same exceptions across that group. In addition, all the change that is except the CU block in yeast mitochondria involve specificity in that labile third position. Finally, it is interesting that the majority of these exceptions are in the mitochondrial genome of some eukaryotes. As endosymbiotic organelles, perhaps they are not as reliant on their own protein synthesis as independent organisms and so have tolerated these mutations. The initial non-functional proteins might be compensated by proteins produced by cell, allowing time for new codon arrangement to evolve. So although there are these few exceptions, the code is still considered universal from bacteria to humans. Dear students, let's now discuss the mutations of genetic code. The sudden change in the DNA sequence is called a mutation. It is mainly responsible for the structural and functional change and for the variation in organisms, which could be good or bad. Even a minute change in the DNA sequence could alter the amino acids to be produced and proteins to be synthesized. Not every individual is similar. In fact, it has been observed that a particular physiological trait goes missing. It happens when the genetic codes get rearranged and deleted during transcription and replication processes. The different segments of DNA get rearranged and deleted during the process, resulting in mutations. Genes are gained and lost in the process, resulting in new physical traits or characteristics of an organism. Let's consider an example. If valine is replaced by glutamine in a particular gene sequence, the individual will develop sickle cell anemia, a blood disorder. Now, wrapping up, the genetic code of an organism depends on the genes received from the parental generation and the way it is produced from replication and transcription. We have learned how the bases from codons and how they direct the genesis of amino acids. These amino acids are units of proteins that form the genetic material of an organism following a proper sequence. There are different types of mutations and are mainly based on the range of alteration. It could begin with a single base pair to a segment of DNA. Common mutations may be either point mutations or frameshift mutations. Dear students, let's now begin with point mutations first. A variation which is caused by the change in the building block and base pair of DNA is termed as the point mutation. An example of point mutation is sickle cell anemia. The sickle cell anemia is a disease condition caused due to the change in a single base pair of the gene. In this condition, the codon for the amino acid glutamate is replaced by that for valine. Dear students, let's now discuss the frameshift mutation. When the reading frame of the genetic code is altered by the insertion or deletion of one or more bases, it is called frameshift mutation. Two frameshift mutations in the CFTR genes cause cystic fibrosis. One involves the insertion of two nucleotides and other involves the deletion of one nucleotide. Dear students, let's now discuss the benefits of genetic code. The genetic code is the set of rules by which information encoded within genetic material, that is DNA and RNA, is translated into proteins by living cells. A genetic code shared by diverse organisms provides important evidence for the common origin of life on Earth. That the many species on Earth today likely evolved from an ancestral organism in which the genetic code was already present. Genetic code enabled direct correlation between DNA and protein to be accomplished. Dear students, this was all about today's lecture. Hope you have understood it well. Take care. Goodbye.